Hello, everyone. We are back again for the live podcast broadcast of This Week in Science. This is the time where we all get together and talk about science and have a wonderful time for a tight 90. And then you know that if we make mistakes or if there's weird technical glitches, things get edited out for the podcast. And so the podcast is the edited version of all of this. You are here for the live show. Thanks for being here. And click all the likes and the hearts and the things that help us with the algorithms and letting people know that we're live and that other people should come and watch us. So share it out with the world. Are we ready?
We're ready.
Yes, I will say.
Okay, I will say. Starting this wonderful show in 3, 2. This is TWIS. This Week in Science, episode number 925, recorded on Wednesday, May 10th, 2023. The science of soap and mosquitoes. Hey everyone, I'm Dr. Kiki, and on tonight's show, we will fill your heads with sperm, assassins, and magic mushrooms. But first...
Don't think I was ready for that.
All right.
Okay. I can do this. Disclaimer. The future is coming. According to some sources, it is already here. Science has gained unparalleled knowledge. Research has found breakthrough applications. And technology has already been set in motion to make significant changes to the lives of humans. Change is coming, and the world as it stands is currently operating under an obsolete model. We are eating wrong, thinking wrong, drinking the wrong water, breathing the wrong air, and are still reliant on fossil fuels despite knowing the damage it is causing. We are still on a money system that incentivizes accumulation over application, is used as excuses for the unethical treatment of humans, and allows life-saving research to languish. Because, insert moneyed interest reason here, while we do tax cuts. Undereducating, underinvesting in the next generation, while raising the retirement age around the world just as working is becoming the most automated it has ever been. But like I said, change is coming, and you'll see it first right here on This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're back again.
Yeah.
Hello. We're here.
We're all here.
Science. We're all here. Yeah. The gang's all here. So let's talk about some science. What did we bring this week? I have stories about diamond funnels, the pangenome, and soap. What'd you bring, Justin?
The preferred sex of space travel. I got a story about what's going wrong with doctors. What are doctors up to that's terrible these days? A possible, it's just a case report. About magic mushrooms and color blindness.
Blair, that's going to be all about you? No. Okay, what's in the animal corner?
No, it wasn't my case study. I can tell you right now. I brought the sperm, of course. You know, sperm's in the show. It's usually my story. And then also, I brought assassin bugs and smelly or not so smelly crabs.
All right. I mean, just listening to this lineup tonight, I am. This is a talk show. Yeah, this is a talk show, and I'm finding myself speechless already. So let's get moving into all of this fun stuff. As we jump into the show, everyone, I want to remind you that, yes, this is a podcast. So if you have not yet subscribed, find us wherever good podcasts are found. Look for This Week in Science. We also live stream weekly on YouTube, Facebook, and Twitch. Again, look for This Week in Science. On Twitch, we're TWIS Science, and we're TWIS Science on the Instagram and the Universidon and other places. But if you want to watch live, it's Wednesdays, 8 p.m. Pacific time. That's where you find us. And if you just want to find all this information, it's at our website, twis.org. That'll help you out a lot, I think. Move into the science?
Let's do it. What do you got?
Yes, I was seeing that. I was waiting for the ready. Let's get ready and go.
Let's go.
And go. Okay, so once upon a time, Einstein was pondering the world. He came up with the theory of general relativity, which we keep finding no evidence against. General relativity. It works. That's great. But he was really, really bothered by spooky action at a distance. He was very bothered by quantum entanglement, and
but despite the fact that he discovered it.
Yes, he discovered it, but he didn't like it. Yeah, he's like, oh, everything seems to go this way, but I don't like it. And he liked the idea of like local realism, that everything was connected through localities, and that there was a connection between the macroscopic and the microscopic and the quantum, and that it could all be connected, which we haven't really been able to connect the macro world with the quantum world yet. That's not happening. But anyway, there was another researcher later in the 70s who came up with an idea. He wanted to study what was called, actually, John Bell, it was the 1960s. He wanted to settle the question of the idea of this locality versus entanglement that could be active across space and time. And so there were random experiments that were performed, not random, but planned experiments that were performed by Bell on two entangled particles at the same time. And if they were doing the same things, then everything was or if they were doing things the way Einstein planned, then Bell was wrong. And if they were doing things the way Bell thought, with what he calls Bell's inequality, then everything's great. Anyway, these are concepts that you can find out about in physics books and everything. So, suffice it to say, experiment after experiment after experiment has been run to kind of repeat these Bell experiments over and over again, because really a lot of assumptions have had to be made through the years to determine whether or not the results are accurate. And one of those is that usually the researchers are limited by space. So where they put their physics measurement experiments and how far apart entangled particles can actually be and how it all works. And so there have been some very, very great experiments that have supposedly settled the disputes over the years, but some researchers just publishing in Nature this week have really reconfirmed the results. And in this reconfirmation of the results, they have shown that semiconducting circuits operate according to the laws of quantum mechanics, even though they are not microscopic quantum objects, they're macroscopic quantum objects. So they showed by taking these superconducting circuits and separating them by 30 meters, they decided that this was enough space between their measurement devices to...
That's a good distance.
It's a
That's
good distance.
really good distance. It's a football field.
And it would take out assumptions of like,
I know.
well, maybe photons are involved. Maybe things are moving at light speed, and just the time it takes for the measurement to be taken, actually there's information traveling at the speed of light that's impacting the results. So they have pretty much shown that that is not the case. And so they created this system with two quantum entangled particles 30 meters apart within superconducting circuits, these qubits. And they were able to show that, yes, indeed, at the exact same time, with no light speed travel involved, that the circuits were working exactly as they should be. So in effect, it was disproving Einstein's ideas of a locality and actually providing more support for the concept of spooky action at a distance, and that entanglement can occur and these objects can be connected and have the same movements across space and time instantaneously. So anyway, new experiment, new technology to allow it to occur, and repeating results, but with more significance. So their data, they did 1 million measurements, and their data is highly, highly, highly significant. The fact that it's highly significant suggests that this is going to, with the superconducting circuits, potentially lead to better encryption in our computer communication systems. Maybe it will lead to...
I love how that is often, like, that to me is the silliest reason. I mean, it's the first one they think of, I guess, every time. Because you always hear about, we can encrypt things. Okay, what's the encryption going to be based on? Well, we're going to use a software that structure was created 50 years ago to navigate that. Like, well, then you just ruined the whole thing.
Yeah. And another aspect of this whole study, while it's very interesting and I think exciting for physicists and also for people who are interested in the potential of where quantum entanglement might be able to take us, the experiment itself, it had to be produced in a very specific superconducting situation, close to 0 Kelvin, so very close to these cold temperatures. So this is not real world stuff yet. This is going to take a really long time before it becomes more real world application. But
so what I'm interpreting from this is,
yes,
Einstein was wrong, but he was actually right because he was bothered by spooky action at a distance anyway. So he was right in how he was kind of wrong.
No, he was right. He just didn't like it. He didn't like the answer that he figured out had to be the result. Like this is the biggest crime that has happened in physics is that Einstein came along at before we had the never dying human, which we're still not quite there, right? But like, what would another 50 years of Einstein having a functional brain working on these problems have produced?
Nothing that anyone else could have understood.
Well, no, but he would be able to. I mean, because people do understand. But like, would he have spent the last 50 years experimentally trying to prove any of his stuff? No, he would have moved on and created new things that people are like, gosh, we got to experimentally test this now too. Like he wouldn't have been done.
That's part of it also, is that he wasn't an experimentalist. He was a theorist. And so he did the math and he couldn't put it all together But now we have the technology. People are starting to put things together. And so even though he was bothered about it, maybe he would have kept working on it and working with experimentalists and, helping to get where we are now. But technology had to move forward enough for us to have superconducting circuits that could, have entangled qubits within them.
Well, there's that. Yeah, there's also the possibility that he would have to work with experimentalists because Einstein at the end. Well, actually, I've thought of all of the ideas. It's right. There's nothing else. There's nothing else to figure out here. I guess I just work with the experimenters trying to show examples of it now because I came to the end and it's all correct. That's it.
It's all correct. It's all good. I think
he'd still keep working on stuff.
Yeah, I was thinking about it, and I'd love to hear from our audience their thoughts throughout the show or even later on who they think are the greatest minds that we know of. Because yes, situationally, people can have great minds and not get recognized for it, but people like Einstein could visualize all this stuff in their heads and hold it in their heads. Stephen Hawking could hold all sorts of equations and thoughts in his heads and hold on to them enough to be able to take the time and have the patience to spell it out letter by letter. So, I don't know. There are some amazing minds that have graced this planet. Yeah. I think it's an interesting question. But do you mind, Justin, telling me a story?
Well, let's see. Where we start? Let's start in space here.
Okay.
So, humans have been thinking about life on another planet. Starting on another planet. This whole thing that we got going on Earth, doing that somewhere else. Well, the nearest solar system would maybe take tens of thousands of years to travel to, requiring thousands of generations of humans to be on a spaceship heading there, which represents some problems. First of all, you have to have a decent mix of men and women. It has to be a large enough number that you don't have too much genetic drift or inbreeding or what have you going on there. So you have to start with a decent sized population, and then you can't be bigger than the ship. So at some point, you got to pull straws to see who goes out the airlock, right?
Or shrink humans. Is that where you're going with this? Shrink
They
everybody.
get to be smaller.
I've always been for shrinking humans. I think humans are way too big. You don't need to be this size anymore. We can be smaller. But more likely, we would be colonizing Mars, which is a much shorter trip. And if we did that, we wouldn't necessarily need to have the Adam and Eve progenitor generations going. You could have an all-male crew. You could have an all-female crew. You could have it mixed up. It doesn't really, wouldn't really make too big of a difference there. Except this is researchers from the Space Medicine European Space Agency in Germany. They conducted a study published in Scientific Reports. It found female astronauts much more efficient. They have lower water requirements for hydration. They have less total energy expenditure while in space. They use less oxygen, produce less CO2, and produce less heat, which is a thing that you have to dissipate once you're in space because you can't just have it keep heating up in the capsule. All of these advantages, plus they can come in smaller sizes. Then they also looked at the effect of body size, and so this is measuring, by the way, similar statures. So this isn't just like a tall guy and a short lady and comparing the two. This is of the same stature, of the same height. The physiological differences between men and women are there. One of the things they tested also was exercise, because you have to exercise in space. In space, you have to, you do this countermeasure stuff, otherwise the body atrophies. They found that women use less energy, they sweat less. Women sweat 29% less than men? Which means they have to rehydrate less. So why does all of that matter, Justin? Isn't there, all this stuff in space where it's like, once you're there, you're there? The cost of getting payloads to the International Space Station, $93,400 per kilogram. They found that on a 1,080-day mission, a 4-member all-female crew would require 1,695 kilograms less food weight. Now, the water might be recycled. The oxygen systems on board might be about the same, so you don't take weight off of that. There's some packaging difference also in the food waste, but still, just with the back of the calculator, 93,400 per kilogram, 1,695, you end up with $158 million savings just in the gender of the crew. Also, it frees up 2.3 cubic meters of space from the waste packaging of the food alone, which we're looking at.
With. Yeah.
Yeah, so that is 4% of the habitable volume of the Gateway Halo module for NASA's proposed lunar orbit space station. So if 4% of your living area is trash, it's quite significant. That's quite significant. I mean,
plus, women never poop, so you don't have to worry about that. So, I'm kidding.
That I didn't know. See, that's why I tuned into this show.
I think one of the things, though, I learned that when I was in Huntsville, Alabama, I learned that instead of sending up ice to pack a whole bunch of the food that is perishable, they pack it in ice cream. So I wonder if they would have to pack more ice cream. Because
fun fact,
women on the show.
Fun fact is they don't actually take the space ice cream. Never goes to space.
No, not the
dust,
not the dusty stuff, but real ice cream, because it has it's able to hold. It has it would be a high heat about, so they can hold the cold in for a while. It's able to keep.
It's also very malleable. I would imagine,
malleable
so
and delicious, and so it's not wasted.
So yeah, relative
for astronauts. Yes.
Relative difference between the male astronauts and the female astronauts on similar stature across these metrics is range from either a 5% to a 20% less comparable usage of energy or production of heat or what have you. Then compared to the 50th percentile stature for U.S. females, 1.6 meters, so that's about 5'3" to 5'4", somewhere in there. They were even better. So they were 11% to 41% better on the comparables. This translates into less use of oxygen, production of CO2, metabolic heat. Well, you know that what was that terrible mission where they're like, oh, we got to hurry up and get back to Earth before we run out of oxygen.
If it were all women, it would have been a much different situation.
It would have been like, oh gosh, it looks like we're gonna have to get back to Earth with enough oxygen for another week up here. So it's fine, we got this.
So pretty soon you're gonna be saying, I'd love to be an astronaut, but I'm too tall.
So there is already. So you can already be too tall to be an astronaut.
That's true.
So interestingly, they don't have a weight. They don't have a weight listed as like, oh, you're too heavy.
Well, that makes sense because it's more about fitness, right? They probably care more about your fit test than they do about your weight.
A body mass index or anything like this as a qualification.
Well, BMI is
a max height
anyway. So. And
there's a minimum height. That minimum height is frighteningly close to what that 50th percentile female form would be. Yeah. So they might need to drop that one because, especially as we're talking about now something that's going to be tighter quarters than the ISS in this lunar mission, this lunar space station. shorter is going to be better in space. The shorter,
Well,
the better.
they're gonna have to build things differently then, because I think that's part of why the height, there's a height minimum, right? Is so you can reach controls and you fit properly in the seats and all these sorts of things that maybe they're gonna start making everything smaller.
Yeah. They need to start making the space. Wait, isn't there a problem? Like they were gonna do a spacewalk, but then they realized like they only had a couple of the spacesuits that would fit women.
The right size.
Yes.
Yeah,
Mm-hmm.
like you need to make all those smaller. You need to lower your height requirement because the ergonomics of working in space in these tighter quarters also is going to be an issue. This is a whole other benefit, which is you've got smaller work areas now. You can't have big, tall, lanky people going around bumping into things. You need smaller people in space.
Awesome.
See, I assumed this story was going to be all about this, like, well, women are better at multitasking, or also some sort of weird societal thing. But this is all about size.
Our metabolism. It's all about our metabolism.
It's metabolism.
Yeah.
There's also so there's also anecdotal stuff that's not in this report here about the eye problems that astronauts have being pronounceably people can say the words being worse in men.
Right.
So for whatever reason, they found that the female astronauts do not have the same amount of pressure buildup behind the eyes that's causing some of the vision issues. So that's another thing. They don't know why that is yet, but that's a physiological reason for potentially having all female crews. And then there's just the obvious. There's just the obvious, which is what the study we just did. Physiologically, they're cheaper to send to space, and if they also can use shorter astronauts, then that's also a benefit.
So now it's instead of a cheap date, you're a cheap person to send to space. Okay.
And if you want to get it slightly misogynistic,
no,
if you need that before we go,
no, I want to.
I will just say
sperm now.
I'll just say I'm going to be slightly misogynistic and say having the big strong man, it's weightless. Yeah, you don't need anybody to help you move the couch. And if you need a tall person, there's nothing you can just push off the floor and you can get to the top shelf, regardless of how tall you're starting. Right? So you don't actually... And one last misogynistic piece, there are no spiders to kill in space. So men kind of like, we don't... If you're not reaching for the tall thing, killing the spider, or helping lift the heavy thing, what good are you?
It's a great question. Especially since we're doing all sorts of work on, replacing sperm, making sperm. But Blair, you had some interesting work about biology of sperm.
Oh, yes.
You want to talk about it?
Yeah, this is a grant and award announcement from Michigan State University. So I don't have results to share with you.
Wow.
I just have a really quick announcement of a study that's beginning that I think is really interesting. So we'll have to have a to be continued when this is released in I don't know how long. But did you know that these small African fish called electric fish or elephant fish, they have sperm that don't have tails. They have no flagellum.
How do they get places?
I don't know. But so there's this $1 million grant from the National Science Foundation that has been awarded to Michigan State University to study these fish and their tailless sperm. The hypothesis is that the reason they don't have tails on their sperm is to save energy. The idea is that they have prioritized the energy spent on their electric fish activities, which is basically they live in these murky African waters. They're so muddy that they rely on electric charges to find each other. And I think we even did a study on the show about them and the way that they talk to each other through electric charges. And it's truly fascinating. But both the pulses of electrical output and the brain power needed to analyze the pulses require a lot of energy. So the idea is maybe this was the trade-off. You get your electric communication, but you lose the tails on your sperm, which seems like a really weird roll of the dice evolutionarily that worked out, but hey, maybe. And so the way that they're going to test this is by isolating the genes that are related to sperm tail development, insert it into a species of fish that has flagellal sperm, so that they develop sperm without a tail, to see if those tails disappear. So then they find that this is the gene, and then they want to be able to measure respiration in both the electric fishes and the non-electric fishes to see if there's a difference in energy output with and without sperm tails. So that's kind of their beginning theory to see why this is happening. Because also, we don't know how it works. How does the sperm get there? Which, this is the other big overarching question of this study, is like the expectation is sperm is cheap, eggs are expensive,
Right.
sperm move, eggs really don't. So
Right. Yeah.
So that's the way it works. But how do these little balls of sperm without tails get to the egg? And if so, is there a different type of investment in their sex cells in this species versus other species where there are sperm with tails? Does that impact the way the males and females interact with each other and how they treat fertilization and reproduction in general? It's a really big question. And on top of that, then you kind of have to ask, like, okay, humans have assumptions about how males are programmed to fertilize and spread seed and all this kind of stuff. And females are passive recipients. But in this case, if it's not like that, then there's a fundamental biological assumption that we are making potentially societally for expectations for males and females as well that could be based in nothing, basically. So this one little study on what happens when you remove sperm tails could have implications for biology in general, which I think are very interesting. So
yeah. So my big question here, though, is why the sperm tail is there to move the sperm to the egg, because the egg doesn't move, right?
Right.
But we know throughout the biological kingdom, like pollen doesn't have a tail,
Right.
does it? Like there are broadcast spawners
Yes.
who just things go out into the environment.
You're
and
right.
they're
So like, do
lucky
they
if
just produce so much? Like, is their ejaculate so full of these little sperm balls that it doesn't matter that they don't swim?
Right.
Yeah,
They're
do they
they're
deposit
spreading
it
through the
very
universe.
locally?
Right.
Yeah, right? Does it not have as far to go? Like, are the females' eggs being released much closer to the cloacal opening? Like, what is happening? I need to know more about electric fish sex right away. So, Michigan State University, please
Right.
hurry. Release your study. I will conclude, to be concluded, on This Week in Science.
To be concluded as Blair is satisfied in understanding more about fish sex.
Yes, and tailless sperm. What even are you? How do you exist?
It's just a cell. It's a thing. Right. What? Yeah.
How do
okay.
you do what you do? How has evolution allowed this to persist? That's what I want to know.
Because it worked and it hasn't messed up yet.
Yeah.
Yeah. All right. Moving on from fish sex and tailless sperm, let's talk about diamonds. Diamonds, we know, they are not all over the world. I mean, they're all over the place. But the ones we like to use and make into jewelry and stuff, they're in these deposits around the world, and they're usually in what are called kimberlite eruptions, which are these eruptions from the mantle of the earth. They come up through the mantle and out to the crust of the Earth. And researchers want to know more about how the internals of the Earth work to figure out why kimberlite deposits are some places and not others. What are the forces at work and how are things happening? Well, some researchers just published in Nature Geoscience on their supercomputer modeling. So they haven't actually gone and looked for these things, although other people have done a lot of work already. And what they found is that there's a lot of data out there, right?
Yeah.
And so they found through their supercomputer modeling that it matched very, very closely to where a lot of these kimberlite eruptions have taken place. But what they have found in looking through the history of what occurs is that there seems to be a plume or a pillar of heat that comes up through the mantle. And they think that there's this pillar of heat that drives up toward the surface and actually ignites these kimberlite eruptions. And so it's a matter of finding how the heat is being transported up from the deep earth to the kimberlites. And they were able to determine that these mantle upwellings, what they call them, and they really, really like the term pillar of heat, is what connects the earth deeply into the surface. And they were able to successfully capture these kimberlite eruptions in Africa, in Brazil, Russia, and also in the United States. Some of the eruptions that we know of in Canada were not discovered, were not predicted by this model. So they think that there may be other mechanisms at work. But they've been able to predict kimberlites back to 1 billion years of mantle movement, of this energetic cycling of heat from the inner core, the mantle up to the surface, and additionally, the reconstructions of tectonic plate movements across the surface of the planet.
Wait, are you indicating that Canadians have diamonds that they just haven't been telling people about?
No, they've been telling people about them. They didn't occur by the same process. They think that they occurred more by a process called plate subduction. So when the one plate moves underneath another plate and causes a lot of heat to occur. But in this particular case, it is these upwellings that from the mantle, from within the earth, that we have this movement that's hot enough to be able to create ignition, that's hot enough to create the situations that are necessary for the pressure and the heat that produce diamonds, which are one of the hardest substances on the planet.
Yeah. So my first thing is Canadian in the chat rooms. Justin, shh. I knew your economy couldn't run on maple syrup. I knew you were hiding.
Hiding the
thing that first jumps out is like, okay, so where do we send the submarine collectors? Because when we talk about the diamond mines and all of this, that's nowadays. Initially, some of the best diamond finds that have ever been collected were just picked up off the surface.
Right.
But we don't need any of that. We can make diamonds in labs.
Now we can.
We can.
That are actually better quality than the things that you pull out of a diamond mine. This is really interesting for geology's sake, I think.
I still want a submarine to go down and pick up some big diamonds. Now we have funding.
Ecological disaster. We
have funding.
We just sell lab diamonds, please.
But that's another question is, these kimberlite locations, that's where we need to be looking. And so they are very specifically located. And many of them are luckily in mountains, on tops of the continents, and on top of the plates, and maybe there aren't as many underneath that we need to be worrying about. Their models have also predicted undiscovered kimberlite eruptions in East Antarctica and the Yilgarn Craton of Western Australia. So there are other places that still may be mined that have not been determined. Yes. But yeah.
And the value of diamonds is all manufactured anyway.
Anyway, and
Because,
yes.
again, geologically, very interesting.
Fascinating, yes. I totally
From a diamond
agree.
selling perspective, I disapprove.
But from a geological perspective, this gives us much more information about how things are moving to the surface, the heat movement that is occurring, and why, different blobs of heat move in different directions, and where they go and why they go there, and why different minerals are formed. And yeah, so
Absolutely.
lots of
And that
interesting
has ecological implications on animals that depend on the plumes or columns of heat or whatever. There's all sorts of cascade of effects of understanding these dynamics, which is very interesting beyond just shiny.
Lot beyond
the
just
value.
shiny,
The
for
value
sure.
of diamonds question thing is kind of like, I don't think anything has value that doesn't have a research output. So
let's just get to the science. Yes.
So I'm in full agreement about that on every subject that doesn't have a research result.
Yeah.
Stem
Speaking
cells?
of which,
Yeah.
this is okay. So, there's a lot of hype in science, specifically around stem cells. Boy, stem cells have been this panacea of curative things that can be done with them, and we can cure all of the diseases, and we can have all of these therapeutics and everything. And where are they? Well, they're not here. So, why?
Yeah.
So a couple of the reasons. One, which is it's probably the lesser of them, but it's that stem cells can proliferate. They can proliferate as any type of cell, creating fresh tissue without disease. You can regenerate with it. You can take these progenitor cells and place them into, a liver or a lung, and they can rebuild liver and lung tissue, and fantastic. Problem is, partly is that they can keep proliferating. They don't always have the off switch in research that's been done here. Also, sometimes they make the wrong tissue. So, you go in to do a hair transplant, and then a tooth forms, right? Or you do a tooth replacement, and it gets this hairy tooth thing happening, or whatever.
That's baleen. That's fine.
Yeah. So a lot of that, though, is probably has direct knowledge-based solutions as well as cell management. In a lot of the research that has produced either runaway or wrong tissue, there's all sorts of questions about how that stem cell line was managed to begin with, whether or not they started differentiating before the experiment took place. And then we're learning so much about cell signaling that learning how to turn off the grow factors. These are things that are already within grasp of being able to handle. Plus, enough experiments been done to know how to use these cells better. So there's that. The other problem is they get rejected. They get rejected by the body and destroyed because of something called human leukocyte antigens. These are markers found on most cells that the immune system uses to say, hey, that's one of us, or, oh gosh, that cell is foreign, and we need to call the immune system and have them destroy it. So these markers. Okay, so then you just match markers with somebody. The problem is there's tens of thousands of these different markers amongst the variety of humans that exist. And you can get really close and have it be effective. or it can be very different because the alleles are just not right, and then the body goes, nope, that doesn't belong here. One solution is you can engineer from a patient's own cells, these pluripotent cells, and then use those on the patient. Now, you're talking about something per patient to even get these started, hundreds of thousands, if not millions of dollars per patient, and so it's not a mass therapeutic. The other idea that's been floated is you could create a bank of stem cells with all these variations that humans have, and you'd have millions and millions of lines of cells that you would have to keep in this bank and have at the ready to go send off to somebody to use, and is unfeasible, to say the least, regardless of the intentions that you have going into that. So now what we have is, okay, how do we get a universal stem cell that can be used for anybody that ignores this human leukocyte antigen marker issue? And it may have just been accomplished. This is researchers at Sana Biotechnology, California. I think they're based in Washington, but this is out of their California lab. They have made a significant breakthrough. They have created stem cells that they're calling hypoimmune-induced pluripotent stem cells. They have tested them on mice and rhesus macaques. And based on the results that they've put out, this is published in Nature Biotechnology, there was no reaction from the immune system. There was no attack on these stem cells or the cell isolates that formed after. In one of the experiments on mice, they had created a pancreatic cell that was, and these mice were modeled to have diabetes. It improved their condition and didn't get attacked. They did something really interesting here too, is they took a wild type stem cell that would get attacked by the immune system, so that you have all of the activity, the cytokines or whatever, they're going in there and attack that are designed to, the immune system's brought all the artillery in to attack the cell.
Yeah.
And of course, those got wiped out. But they also did it concurrently with their cloaked stem cells, with their universal donor cells. And despite being in the same area as the cells that were getting attacked, they were left alone.
This is amazing.
This study, which is kind of obscure by title, where's this? This is it's not saying, "Hey, we did it." It's hyperimmune-induced pluripotent stem cells survive long-term and fully immunocompetent allogenic rhesus macaques, meaning that they did not use immunosuppressive drugs that prevented the immune system from putting forward the attack. And these were not type matches. These were not matched HLA so that they would not.
Yeah.
Yeah. They were unmatched.
They were rhesus macaque pluripotent stem cells, but not matched to
They used human.
individuals.
They also used human in the rhesus macaque. And in the mouse model. They were completely wrong, right? Didn't get attacked. And there was no immunosuppression of the subjects. Those two accounts right there mean there's this stack. Okay, there's no stack of research papers anywhere on the planet anymore because everything's electronic. But I'm picturing this teetering stack of research that's been done in lab conditions where they say, here's the different things we might be able to cure with this that have not been able to be implemented, that have not been able to be clinically trialed, that are not going to have a mass therapeutic potential because of the issues we were talking about. There's too many factors to get by the immune system. These also lasted, I think one of the tests was 40 days. It was the longest test. There was a 16-day in the initial portion of the study, which was the length of that one in the macaques, where they said, okay, the cells are still here. They're still...
It's not days, weeks. I'm looking at the
I'm sorry,
figure.
16 weeks. 16 weeks.
40 weeks.
Versus 40 weeks.
Yeah.
Was the final one, correct. And so their long-term survival is,
and I think that
pretty intense.
And I think that's the interesting point, and the questionable aspect is, when you put in a pluripotent stem cell, one of the concerns is whether or not it's going to become tumorigenic. So is it going to go in and just start dividing and form a tumor? And one of the things that can protect a body against that result is the immune system attacking this foreign cell. So I think one of the big questions is going to be moving forward, determining whether or not these implants can be safe. Not just that they are across the board. You could give them to everybody. I think that's awesome. But then, how is this going to impact over the long term? The growth of the things that you're trying to fix, the development, whatever it is. Yeah,
that's one of the three. That's one of the three obstacles to stem cells. Tumorigenic, immune system rejection, and the necessity of immunosuppression long-term in a patient. So, immunosuppression, long-term patient, gone. Rejection by the body, gone.
Yeah.
And what you end up with is still the tumorigenic effect, which, from my reading on this, a lot of that has come from stem cell line management.
Yeah.
Now the proliferation that can't be stopped in some cases, is also, that depends on where it's being used, maybe whether there's already in that area the stop growth signals aren't present for whatever reason, that they're not reaching the stem cells. But for the ones where it was, I can't remember the terminology for it, where it grows the wrong tissue.
Right.
Those they think is that's lab condition. That's stem cell line handling issues that have largely been sort of figured out over the years why that takes place. And we know enough about cell signaling, I think, that we'll be able. But a couple of the huge hurdles may just be completely out of the way by the good folks at Sana Biotechnology.
Ooh, a company that will sell and license their technology to everyone. But it's great. This is wonderful. It's fantastic. Yeah.
Yeah.
Yeah. Yeah, I love it. This is the kind of thing that we need to be working towards. Another aspect of things that we need to be working towards is not just having a human genome project that has basically just been like, oh, look, it's like one person's genome, or putting everyone's together and just saying, look, it's a puzzle and we put it all together and this is all of our genes. Researchers last year said they finally completed a complete version of the human genome. Right?
Wait, I thought we did that a long time ago.
Like in the 90s.
Right. But last year we talked about this. It was adding in new things that hadn't been. It was the most complete version
at the time
of the human genome ever. So it's still just like, hey, it's the human genome. This is our genome. This is the one genome. And this represents everybody. When we know that there are all
whose genes is that?
Yeah, so we know that there's like all sorts of diversity in the human genome generally, right? Lots of people have mutations. We have all sorts of fun things going around. And researchers have finally come together. There were 4 studies published in Nature this last week that describe their construction of what they call a pangenome. So it's the next level in human genome understanding. It's 47 different individuals from around the world, but of course it is not representative of all of the different groups of people around the world. And it has taken into account many differences between different groups of people. So it's actually showing us how our evolution and adaptation has led to certain differences within the genomes that we have present on the planet today. And so this is a big project that they are continuing to work on. They want to continue adding more and more people to this. They're in talks currently with indigenous peoples from around the world to see about including more indigenous representation into this project. But yeah, I think it's a great step forward because it is taking that idea of just like the big average blob of the human genome and suddenly starting to look at it as a much more diverse construct.
Can't they just source all of the 23andMe
Well,
information?
That would be almost essentially exclusively the Americas.
Sure,
it's
but
not.
There
It's not a European.
There are different brands that are popular in other countries too. There's
Sure.
and yes, it would skew towards a certain type of person that would send their information in. But I'm just saying that this stuff, there are DNA samples of humans out there in large supply. That I feel like there's an opportunity there to just source a bunch of it.
For sure, but a lot of that is, like I said, it's single nucleotide polymorphisms. So it's not entire genomes. It's just little tiny snippets of genomes. And so, the shotgun sequencing that's been done to be able to put things together and piece things together, they're trying to advance this technology to allow it to be faster and better and more representative. But one of the cool things that they've discovered is that there's a lot of interesting duplication within different individuals, that where we have certain segments, genes, that have been reversed or duplicated multiple times. And so some people have 1 or 2 copies, and some people have like 4, 5, 6, 7, however many copies. And so now the question is, how does this represent the area people come from, the evolutionary lineage that they descend from? What are the how does it impact protein function? And they're also starting to look at things like the epigenetic aspects of the genome also, because they're able to start being able to look at different loci that are responsible for the twists and turns of DNA that hide some genes until they're accessed for transcription into proteins, or even during this process called imprinting, which turns off one chromosome and turns on another chromosome. Which ones are actually the ones that go on to produce the proteins that impact who we are. So this pangenome is going to give us a lot more information about the crucial loci within the genome, and for different lineages of people to be able to determine how different genes are expressed in proteins and how it goes into making us who we are. Yeah. Mm-hmm. Anyway, yeah, they want to end. They need more people. So more genomes. So far, 47. Which, when you think about science and sample sizes, it's very small. Yeah. But it's better than what we have.
It's better than the one.
Yeah.
And then the other thing to toss onto this is that this is also still just scratching the surface then of genetic difference. Because, all right, you've got this, some people call it DNA like a blueprint. It's more like the shelves of at a hardware store, right? These are more like the ingredients that can be applied. Where the actual application takes place is in the transcriptome, and how genes are edited, and how mRNA messaging and regulating takes place at this next whole other layer, right? Which can have ethnically connected differences. I don't think we ever talked about this on the show, but there was this study that was looking at schizophrenia. And they found this really strong signal with some types of gene expression that were elevated, that were repressing neural activity and what have you. What was interesting is because they had included people from diverse ethnicities in this study, they found that was only true in the European cohort. That there was almost the opposite being seen when they were narrowing down this one portion of it, and I think it was the African population, meaning that there was more than one pathway to
The
to
same
the disease,
effect. Yeah.
to the effect, but that it was different micromanagement within, So the genome is. Again, it's a list of ingredients. How they get applied is a whole other layer that this can give insights into.
Yep. And this is one of the steps on the way to being able to do that. Thank you all for joining us for This Week in Science. We love that you're here once again on our weekly journey through science news, talking about all the things that we love to talk about. If you're enjoying the show, please share it with a friend. We love it when you share the science. Don't keep it to yourself. Share, share, share. Also, head over to twis.org. Click on our Patreon link if you are interested in supporting the show moving forward. We do appreciate your support. We really can't do it without you. Thank you for your support. And now, on This Week in Science, it is time to have Blair's Animal Corner.
With Blair!
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I have tool using assassin bugs.
What?
Terrifying, I know. This is from Macquarie University in Australia. They have found the first example of tool use by assassin bugs in the field. Assassin bugs are a group of bugs that are clustered together based on a behavior that they have. They kill. The way they do that is by poking a hole in their prey with a rostrum and pumping in digestive enzymes and then sucking out all of the innards to eat.
That is
Delicious.
That is so clever.
So delicious.
Yeah.
But it does require a certain amount of sneak uppery and cleverness to be able to grab, poke, digest, and eat. And so, it's very assassin-y, I suppose.
Oh, very charming looking insect, too.
Oh, yes. Oh, yes. Quite charming. And what you'll notice based on these pictures is that they look kind of shiny, sticky, don't they?
Yeah.
So they are, specifically in this study, they studied a group of assassin bugs in the Goreraduvius genus. They live on blades of spinifex grass, which is a type of grass that produces a sticky resin. This grass is well known in Australia, mainly because indigenous people would use the resin to bind material together when making tools. So everybody knows about this grass. It's nice and sticky. And so they had an idea that these assassin bugs, since they were living on this grass, might be using the resin. So researchers collected 26 specimens, brought them back to the lab. Both males and females would scrape resin off of grass blades and apply it to the parts of their bodies. Mostly it was on their forelegs. It's on their arms, basically. Then they placed individuals in glass jars after they had applied resin. Then they would add an ant and a housefly and watch. And the assassin bugs would try to capture and eat them. Then they did the same experiment, but before they put the assassin bugs in the jars, they took some makeup remover and they removed the resin off their legs. So they wanted to see if they were less effective at capturing their prey, and they were indeed. They were 26% more successful at capturing prey when they had resin on their bodies versus when it was removed. Conversely, they found that flies placed in the jars with assassin bugs were 64% more likely to escape if the bugs had the resin removed. So just in general, the resin was helpful in the capturing and eating of prey. So their idea is, based on the observations, that the resin helps them slow down the prey, hold on to them long enough for them to be stabbed and subdued. So it appears to be tool use. Now, I brought this because
we want to
calling
question
it tool
this?
use. I feel like we have this debate every time about what is a tool and what is tool use. And I would say that it is based on some of the other things that we've called tools in the past. Like I called a circular blade of grass being used to amplify sound a tool, right? So it's a piece of your environment. They're not manufacturing a tool. They're not doing anything like that, but they are applying it in a specific way to a specific place to be used for a purpose. What do you guys think?
So I would call it chemical use.
So is a chemical a tool?
But the chemical of this it would be a tool in this case.
It's a great question, but I would put it a little past tool use. It's one thing to use an object, a physical object, to do another function. That's a tool. When you start getting into using a compound, now I'm like, whoa.
But we're talking about
that seems like even more like fascinating to me than just using a stick to. Like, I think that's more. I think it's more impressive than an ape using a stick to get a termite. Is saying, you know what? To catch my prey, I need to be stickier. I'm going to coat myself in this.
Yeah.
chemical.
So is it? You talk about a primate using a stick to get termites. That's intentional. And many times that animal has learned to use the stick, figured it out, or been taught by watching others. And so there's cultural transmission.
Right.
But is it something that if you just gave the bugs nothing, would they figure it out? Is this something that how do they know
like
to
if you
do
put
this?
them on grass that didn't produce resin, and you put
you
put a jar of resin over to the side, would they be like, I'm not sticky enough?
Dabbing up a little bit before.
Yeah.
That's the question. Kiki, you're exactly right. That's what they need to figure out. Is this just a byproduct of where they live, or is this something they are intentionally seeking out and using? You're totally right.
Because I feel like organisms, animals, from the single-celled to multi-cell, like there's things that are happenstance that are uses of stuff in the environment because you've evolved and adapted along with those things. It's not, and it's used for a purpose, but what was the intention and how did that occur? I don't know. I feel like
koalas eat eucalyptus, and eucalyptus helps them to secrete an oil that makes them kind of bug repellent. Is that tool use, or are they just benefiting from the fact that they eat eucalyptus?
Yes.
And that is a byproduct of that.
And they started eating more
I'm
eucalyptus
less impressed with this bug now. Now that you put it that way, I'm a lot less impressed.
No, I think Kiki is dead on here. You have to figure out intention. You have to figure out, are they just hanging out on grass that's sticky and they're benefiting from it by chance? And so evolutionarily, assassin bugs that hang out on grass survive and have more babies. And so that is a tendency that is ingrained in them. Or are they going, oh, gotta lather up, time to go hunting?
I need my sticky stuff. I'm going to go catch some bugs.
Question. You're totally right.
Yeah. I want to know this. They need
Is
more.
it tool use?
I need more research.
Yeah. And now moving on to crabs and their sense of smell. So this is a study from University of Toronto looking at ocean acidification. I will give our listeners who are less than familiar with ocean acidification the, 3-sentence version. But basically, when we burn fossil fuels like coal, oil, and natural gas, it releases carbon dioxide into the atmosphere. Some of that is absorbed into the ocean. That changes the chemistry of the ocean. It makes it more acidic. That doesn't make it like acid. It just makes it less basic and more acidic than it was before. And there's all sorts of kind of domino effects from that. Animals that need calcium carbonate in their bodies that reacts with the ocean in a different way, all the free hydrogens that weren't there before. And then there's less free calcium carbonate for them to grow, so they have less strong shells. There's all these issues. We have talked on the show about how it impacts other animals' chemoreception. There's a bunch of different ways that ocean acidification can impact animals. So I have a new one. So University of Toronto wanted to see how climate change and specifically ocean acidification could have an impact on one of the largest fisheries in the world, Dungeness crabs. They are valued at more than $250 million annually as of 2019. And so they are very common. If you are in the United States, there's Dungeness crabs on a lot of menus, specifically here on the West Coast. They're quite prevalent.
And delicious.
And delicious. I agree. Well, it turns out that ocean acidification might be causing a problem with their sniffer.
Oh.
And if you think about it, you might not really have an obvious kind of place for a crab's nose to be.
Yeah.
It's not quite prominent, right? So crabs have poor vision, so their sense of smell is really important in finding food, mates, suitable habitats, and avoiding predators. They sniff through something called flicking. So they have antennules, basically antennas, that they flick around and they detect odors. Tiny neurons responsible for smell are inside the antennules. They send electrical signals to the brain. So it's actually not that different from our nose. Our nose on the inside, the squishy stuff, our nasal turbinates, are a bunch of nerves that send electrical signals to our brain to tell us what we're smelling. It's a very similar process. It just looks different. So what they find is that their flicking rate increases when they detect an odor they're interested in. So, but in crabs that were exposed to ocean acidification, the odors had to be 10 times more concentrated before they started flicking interestingly in an important smell. So they're flicking less. They also found that they have less sensory neurons than crabs not exposed to ocean acidification. They tested the electrical activity in their sensory neurons to determine if they were more responsive, less responsive. They had fewer receptors, and their sensory neurons were physically shrinking by as much as 25%. So this is a which came first question. Is it shrinking because of ocean acidification, or their hypothesis is that
shrinking causing the ocean acidification?
No. They think that it's shrinking because of lack of use. So because the electrical activity is dulled, they're not using the neurons, and it's like a muscle. You don't use it. I'm not going to put energy into making these things anymore. So they end up being smaller.
Use it or lose it. Yeah.
Yes. So all that to say, they are responding to smells less, and the neurons that tell them about smells and what's important are not responsive. So their smeller's all messed. And so there's implications for that. Obviously, if they can't find food, that's going to impact them. If they can't find mates, that's going to impact them. If they can't smell predators nearby, that's going to impact them. And so, of course, this is a problem for crustaceans throughout the ocean. I would assume that since they have similar structures, they will be impacted similarly. To kind of raise the economic question, you have this big Dungeness crab market. You also have Alaskan king crabs. You have snow crabs. You have all sorts of crabs that are part of fisheries that have an economic impact. But beyond that, crustaceans are a huge group of animals that are detritivores. They're the cleanup crew in the ocean. If we start losing detritivores, we are in big trouble. So this is important to know.
It's part of the whole ecosystem turnover life cycle. It's how it all works very smoothly. We need to have the janitors.
Yeah, exactly. They are so important to the ecosystem because there's also a certain feedback loop with that. The chemistry of the ocean will continue to change if they can't remove waste material from the ocean. Visibility, turbidity will change. The ability for certain nutrients to be free floating will change. It's all impacted. It's just like on Earth or on land. If you didn't have animals getting rid of dead stuff and poop, the ecosystem will collapse.
Yeah, you'd have piles of dead stuff and poop.
Yeah, you'd have noxious air. There'd be all sorts of problems.
So my brain might have checked out for something here. Was this observed in lab conditions, or are these the CO2 conditions of the waters where they retrieved these crabs?
So it was both. So they were specifically pulling crabs from the ocean and testing electrical activity. They were also raising crabs in the lab and testing what happened. So they were kind of trying to see not only what's happening, but what's the mechanism. So there were multiple steps, but
the reason I was thinking this was if it was just wild, and so this might not be correct. This might be an example of human-caused selection, because the way that we fish for crabs is through their sense of smell. That's how we catch them. We put down a cage of crab food, and they smell it, and they go in the crab cage, and then they can't get out. So if the bad smellers, the ones who didn't where's everybody going? Wherever they're heading. It could be we're doing how we do we have done change the average size of fish through overfishing.
Right.
We could be changing the sense of smell of crabs by smell.
Yes.
But that's only if they've done this fully wild.
That's a very interesting point. I think one of the things that they use to kind of point away from that is they cited some other research that was done at University of Hull that showed how ocean acidification disrupts odor molecules. So ocean acidification in a laboratory setting actually does change the way odor molecules travel and the way they bind to smell receptors. And so that's actually something that we talked about with, I think it was with sharks. It was probably almost 10 years ago now. But but because it was when I worked at the aquarium, so it was a very long time ago. But we know that ocean acidification disrupts sensory systems in animals, generally speaking. So it's not a huge leap to go there, but I do think it's an interesting point that there can be other influencers, selective pressures also that could be impacting these.
And it
guys.
could be a combination of all of these things. It doesn't have to be an either or, it could be an and.
Yeah.
So, I mean, and the big issue is these crabs who can't smell, they're not going to be finding food. They're not going to be finding mates. Like their entire behavioral repertoire is going to be messed up. And so it could mess up future populations and the population growth.
Or it could be preserving them. Because here's the other thing.
I know if you go back to the trapping stuff,
if you have reduced, not that you can't smell, but you have reduced range. Now those crab cages that you would have shuffled over to in the old days, now you can't even tell it's there So you keep going about your local business and finding your very local food, and don't go traveling in search of
Or you find
So it could end up being a stochastic survival strategy for the by selection.
Oh, so Justin,
We'll see.
I'm cracking up. I'm cracking open the paper here.
Yeah.
So it's even. I think this refutes your theory because they acquired most of their subjects from a grocery store, which means they were caught. So they
There we
weren't the
go.
ones that don't get caught. They were the ones who get caught.
So they've okay.
They're still smelling. Their sniffers are good.
Yeah,
Yeah.
but so the majority of the of the cases were pulled from the grocery store, and then they put them on these 10-day and then 4-day treatments of carbon dioxide at different levels, and then they kind of looked through all these other things happening. But so they were wild caught by a grocery store, and then they were tested after that. So I don't know.
We'll see what happens with the crabs and other animals and what we do with ocean acidification, because it's still happening, and how that impacts so many different populations.
Another reason. Cut it with the climate change, please.
Cut it.
I haven't really heard a whole lot of the upsides.
No, there's really not
really.
many.
Not many.
Yeah.
No.
Hey, Justin, you want to talk about some studies?
So this is a study about prescription drugs in Americans. They had these researchers. Where are they? This is from Johns Hopkins Center for Drug Safety and Effectiveness. They got a hold of insurance data of 9.1 million continuously enrolled adults aged 19 to 64 from a commercial insurance claims database. And they looked through to see what drugs were being prescribed. They found that 3%, 276,000 of this 9 million, were using Schedule II stimulants. Among that group that was being prescribed, that 3%, 45% were being given a combined use of one or more additional central nervous system active drugs, and were on it for 213 days at least, based on supply for this 1 year study. Additionally, 24.3% of stimulant users used 2 or more additional central nervous system active medications for at least 182 days. And 47.6% also had an antidepressant. 30% filled prescriptions for anxiety, sedative, hypnotic medications. 19.6% also received opioid prescriptions as part of this. And so this is the thing. They also found that once patients started a treatment, 75% of them became long-term users. So what's interesting to me about this study was that all of these drugs that people are taking undergo a clinical trial. Here's a condition that needs to be, that can be treated with this drug. Let's try it. We go through clinical trials. You have safety, effectiveness, all of these, this sort of, here's what you need to know. There are not clinical trials for being on 3 or 4 of these drugs
at the same time. We just know,
concurrently.
Yeah.
These are Schedule II drugs. The reason they get this, FDA regulatory moniker is because they are habit forming. They're very addictive and have withdrawal symptoms and are hard to get off of. Mm-hmm. Each for one.
But then in combo What does that mean?
What does it mean when you're on 2? What does it mean when you're on 3? What does it mean when you're on 4? And so what occurred to me in looking at this was there is... If you were to do a clinical study, one of the thresholds for doing a clinical study trial on humans is that it has to be safe and it has to have a reason for doing it, and there has to be informed consent about the risk benefits. How are these doctors doing this when you could not get a study past good clinical practice to be authorized to do the study? That they don't, each of these drugs, again, there was a clinical trial which informs research informing clinicians.
Yes.
On how to use. How is it that doctors are ignoring that? They don't have clinical trials for these drug combinations. Because essentially what this looks like to me is experimenting on people and doing so in an uncontrolled environment with multiple high addiction, long-term dependence that can result in this. And guess what? There isn't seemingly an exit plan.
So I think, you know what I'm thinking about, Justin, is just to give a personal example of something that's going on with me right now. I was recently prescribed a new medication, and I got an email from a pharmacist at my insurance carrier who said, "Can you let me know which of the following medications on your chart you're currently taking?" There were 25 medications on that list. I am not taking all of those. Some of those are from like 10 years ago when I sprained my ankle and I got diagnosed one thing, or I got prescribed one thing, and then another thing's from when I got stung by a bee last year and I got prescribed something else. And like, it's just, I think part of it is just, at least in my experience in the American medical care system, I think there's a certain amount of throwing up of hands with prescriptions where it's just like, you expect me to do the due diligence to figure out exactly what everyone is taking when, and it's just kind of like, it's just, if they're going to ask me, then I can say, oh, let me look into those interactions for you.
Right.
But otherwise, I think there's a certain amount of like plausible deniability with this, where there's just an expectation that, well, I can't possibly be expected to know all the drugs everyone's on all at once.
And we know that there are certain interactions between certain classes of drugs. And, yes, unfortunately, a lot of the understanding that we have has been the result of people taking drugs in combination and then things happening and you report it to your doctor and then they go, oh, stop doing that, or because they know the mechanism of action of a particular drug and they say this particular drug works on this receptor, so you don't want something that's going to interfere with that or with a particular pathway. So we do know some of those things, and there are definitely those instructions of don't take this drug with that one. You shouldn't be doing that. But yeah, it's a very interesting point that is brought up by this study about the idea that people are using these, and I think the key point, but it is applicable to all sorts of drugs that we take for our mental health, our physical health, everything. But the key point here is that they're talking about addictive drugs,
right,
that are getting mixed in with these other drugs. Yeah, and that in itself is, it is questionable, but we are doing it.
Yeah, it's not good. I think it's Justin, your point is taken that it needs to be paid closer attention to.
Yeah.
But that's my just inkling based on my experience in the healthcare system we have in this country, is that like there's a certain amount of, well, there's too many people with too many different drugs they're on, and we're just not possibly going to worry about all of it.
I mean, there's a certain point where how do you do all those studies for all those combinations?
You can't possibly.
How do you?
Yeah.
So part of it is there are databases of that the doctors are going to refer to for pharmaceutical databases showing interactions and things like this. Okay. But that's not a study.
Right.
it's still, I mean, the thing is, if you don't have a population that you can do the study for, then you don't have a population you should be prescribing this to. Does that make sense?
But,
So,
but
for instance,
if I can push
but-
back,
we have a stimulant. Oh, I can't sleep. Here's a suppressor. Here's one to help you sleep. Oh, now I'm constipated. Here's the second drug. Now I'm this. Oh, try this one in combination.
Yes, absolutely.
It's experimenting on people because they don't have side effect outcomes. They don't have a clear clinical trial that says you give this 4 combination drug to treat. This is the indicator for using these 4, and so you can use it for this long because we studied. And here's what you have to give them to get off of it, or here's how you taper it down. That's
Right,
not
but
part of this.
No, but at the same time, we know that certain drugs work for certain things. And if they don't have negative interactions together and they don't have negative side effects altogether, they can't be managed. And together they improve the life of the person.
Temporarily.
If you are, we don't know. So as a doctor, do you deny medication to people because the interaction studies have not been done, even though we know that there is no specific interaction that's going to be negative?
Because we don't know. Because they haven't done a clinical trial.
So you
Because
don't give people drugs that could help them live better lives.
Correct. So
I don't think that's what
Yeah,
people
I
in the
think that's
medical
problematic.
community are going.
No,
That's
I think the real
Here's-
takeaway here is that as a patient, you need to know that not every combination of drugs in the world has been tested or will be tested. And therefore, you need to be knowledgeable to ask questions and
that's the
pay
problem.
attention to these things and report issues when they come.
I do not like putting it on the patient. I do not like the doctor deciding to experiment. And
we
here's
don't
the
have
other thing.
enough money.
In
We
the
don't
wake-
have enough time. We do not have the infrastructure to be able to
We
test
don't have
all
the
these
money. Do you know how much
interactions.
money is being generated by these drugs?
We don't
Excuse
have
me.
all of
There's
the,
plenty of
it's not gonna
money
happen.
is
I'm
not
just
there.
gonna say
There's
based
a
on-
practically infinite number of
willingness.
combinations of drugs
Yeah.
that you could test.
Yes.
But I'm talking about these are Schedule II addictives. Because this is the lesson from the opioid crisis where patients needed drugs that were supposed to be prescribed by clinical trial, short term, that got prescribed for chronic
Long
pain,
term. Yeah.
long term. And then what happens is, in the United States, insurance is not guaranteed. Your prescription coverage is not guaranteed. So what happens when people came off of their job that had a prescription coverage for the medication or multiple medications that they were addicted to, there was no, okay, now that you don't have our insurance, we're going to put you into rehab for 4 weeks so you can recover. Or go through lifelong, you're now a lifelong recovering addict from this thing that we're not going to pay. Instead,
this
people
isn't-
ended up using street drugs.
Right.
So this is the outcome that I see from getting people on addictive drugs long-term without study and without a safety net for recovery.
I agree with that entirely. I feel like our system is broken in that way, and I think you're absolutely right. We should not be putting people on addictive substances without a safety net. Like our insurance system, the way it works, I absolutely agree with you and take your point here. But that's, yeah. Let's move on to fun drugs. What about magic mushrooms?
So this is not a study. Disclaimer, disclaimer, disclaimer. This is not a study. This is a case report, which was brought by the Center for Behavioral Health, the Department of Psychiatry and Psychology, Center for Behavioral Health, Neurological Institute at Cleveland Clinic in Ohio. And the reason it's not a study, it's kind of interesting the way they called it a case report because it's one individual reporting this. However, this individual is referred to in the paper as a colleague, which, must then, of course, you would assume is a colleague at the Department of Psychiatry, Psychology, Center for Behavioral Health and Neurological Institute at the Cleveland Clinic. Possibly. Or it could be just academic colleague, I suppose, but they refer to this individual as a colleague. The person had noticed when they were using psychedelic drugs, they noticed that their vision improved because the individual, in the case report, is colorblind. Now they're diagnosed with the sort of mild red-green confusion type of color blindness. But they had noticed this enough that they went and created their own trial, along with they utilized what's that test called? Blair, do you know the name of that?
Yeah, I don't remember what it's called, but the dots with the numbers in it.
So it's the dots. It's the Ishihara test.
Oh, okay.
You got this conglomeration of dots of different sizes and slightly different hues. Some will be green maybe, and some will be red. And if you...
This is the one I had to do every time I got a new eye doctor growing up because none of them believed me that I was colorblind.
Because it's so rare.
Because it's so rare for a girl to be colorblind. Yes.
Yeah.
So I had to do this a million times. Yeah.
So this test, if you are not colorblind, you look at it and you see this goofy number in the middle in this. You can say, oh, there's a number 6. That's the number 3. That's the number 2. If you're colorblind, it's indistinguishable. It's just a bunch of dots. Can't see that there's a hidden number in differentiated by the hues. So this individual went from getting what was it? Let's see here. I'll get the right number if I can. 14 out of 21, which is enough to put them in that marginal colorblind. If you're 17 or above, you are considered not colorblind. So the individual took the magic mushrooms and had no improvement.
Okay.
Mm-hmm. Slight improvement. Maybe got one more.
Okay. So far, so bad.
Yeah. 24 hours later, the score was 18.
Okay.
The score remained 17 to 18 for months after the experience. It remained high, and it stayed high for a prolonged period of time afterwards. Now, one of the things the researchers suggest is that the psychedelic-induced phenomena may be primarily from alterations in brain activity rather than any effects of cones and rods.
Yeah, so this is the first thing I thought of, is how my Enchroma glasses work.
Yes.
So you have to wear them for at least a week before they really start to work.
Because your neurons have to shift.
Yes.
Yeah.
So this sounds very similar to that.
Doesn't it? Yeah, so the brain is doing so much of the work. Because this is an example. It's actually oddly used in this paper of Enchroma glasses. Or not in the study, but in this write-up I've got here in front of me. Of Enchroma glasses taking a while to kick in because the brain has to catch up with the separate wavelengths. If the psilocybin induced thinking, brain activity, actually can notice that these wavelengths should have been separated and that this confusion range in the middle needs to be ignored in some way, and then affects that change in the brain without lenses, it illustrates that both are on one level a learning process, because this type of color blindness tends to be that wavelength that's usually like, here's the green wavelength and here's the red wavelength. They begin to overlap. And because they overlap, the brain goes, too hard, call it the same thing. It'll just call that with the one color, and it interprets it the same. So really what's taking place, even though the cones are there, now the people who always push back and think the Enchroma glasses are fake, don't think that this is real, there's people with the severe cone missing, not the overlap, but they're missing some cones. Those people with the severe, yes, there's no help for you. You're colorblind and are going to stay that way, and no treatment will help because you got cone problems. For a lot of color blindness, it's overlapping wavelengths for the milder cases. For those, right? And so the glasses work. For those, apparently, at least in this one case report, showing that the brain itself can overcome this. In this case, with the use of magic mushrooms. But, that's...
But the question is still, like, why would the mushrooms lead to this learning process, and why would the change in vision occur in the first place? And, what is it about the magic mushrooms, the psilocybin, that would initiate this alteration? And
psychedelics, too, because I guess
of course. It's changing stuff, but why is it changing the vision, and what is it? What's the brain learning, and why? That's an interesting place.
So apparently the research the colleague of the researchers initially had made this discovery with LSD.
Oh, so they've done this before.
Oh yeah.
And I love it's a case study. This is an individual. We're not talking about like
they're highlighting it as a reason to do more research into this.
Pilot. Let's take a look.
Yeah. But in these psychedelic drugs, when they talk about the doors of perception are open, the mind actually becomes very perceptive to things that it normally is not.
Right.
Paying attention in different ways.
It's like turning off your filter, basically.
Big time.
Yeah.
Actually, there's something called tracers and trails where, a car drives by at night and that tail light is like a tail coming off the thing almost. It stays, the brain is still processing it even though the car went by. So somewhere in there, that processing of the information from the eye is noticing that you have two different signals that are overlapping and begins to separate them in the mind, allowing your eye, which is already doing the same work it's always doing, allowing your mind to see what the eye is picking up in such a way as reversing the effects of the color blindness.
Maybe.
And most vision, the majority of vision is because of the brain.
Yeah.
So our brain is the super important part of this.
Yeah. And then I'm kind of curious, about all this, these artists who were and it's all the colors I can see or whatever. People who've experimented. It's you do this drug, you see all these different colors. I've never seen anything new colors. Now I realized, those people were colorblind. They just hadn't been able to tell red from green before. That's what happened. It's not new colors that they're seeing.
Maybe.
They're just seeing colors for the first time. Now I get it. Now it explains actually a lot retrospectively.
Yes. Suddenly the world is Technicolor. But how is it that our smell impacts how mosquitoes decide to bite us or not? We've
Our
talked
smell.
about
The way we smell.
the way we smell.
Got it.
Right? Yes, not just what mosquitoes see, but what they smell is very important. And we all know that carbon dioxide is one aspect of what we exhale that mosquitoes get attracted to. There are other scents that either repel or attract mosquitoes, and we have wonderful chemicals like DEET, which are also not very safe for us, but we use them because they're great for getting rid of the mosquitoes, kind of. And then we have other, we know things. They're plants. They're things that work. So some researchers just published in iScience this week a study of how some big name soaps, commercial soaps that people use all the time on themselves, how they change our scents and how that impacts mosquitoes. And
can I guess already? Can I guess
you guess?
something?
Well, of course you can guess.
I just want to get because my guess is that the soaps are attractive, not repellent. That these cosmetics or soaps are actually attracting more insects. Because I don't wear any extra anything, right? No deodorants, no, but, and mosquitoes don't like me at all. But my wife, who's has girly shelves
Lucky.
of stuff,
They
gets
love her.
devoured. Absolutely devoured by insects or mosquitoes.
Well, a lot of people use these personal care products involving soap, and a lot of these soaps have plant-related volatile organic compounds that then are, when we heat up, they aerosolize and they get into the air. And yes, human odor is modified by soaps. This is true. And they used chromatography mass spectrometry to quantify how human odor is changed. They showed soap changes your odor. Dun, dun, dun. Big surprise there. We know this. You can smell people who have washed with soap versus people who have not. That's the thing. So now they tested it on mosquitoes to see what they were attracted to. The different soaps that they investigated were the big names Dial, Dove, and then a couple of lesser known Native and Simple Truth. These are highly popular brands in the United States. And so that's what they looked at the way that it affected the attractedness of Aedes aegypti, which is what are the bad ones? Spreads yellow fever, spreads all sorts of malaria, all sorts of fun things. And what they determined at the end of it all is that the majority of the soaps attracted mosquitoes. So there you go. Yes, the majority of the soaps were attracting the mosquitoes. However, there was the native soap tended to repel. And the reason they think it repels is that it has a high content of coconut oil. And there's research that suggests that coconut oil is repellent to mosquitoes. So
that's what I was gonna say is if any of the soaps had eucalyptus in them, so I just mentioned that earlier in the show, that could have an impact on it. I know some soaps have that smell. And so yes, if you smell like a flower, bugs will want to come to you.
I love you.
So yeah, that's the question. Are you picking a smell that's attractive, or is there something else in the soap besides the scent that is attracting the mosquitoes also? Because I also know that like we've talked on the show about how there are genetic links to who is attractive to mosquitoes and who is not. So
there is that as well. Yes.
Even two people who wore the same soap might have different impacts of what's biting. But I'm curious if it's the scent, if it's a chemical compound, if it's all of the above, if it's how clean it makes you.
It's no, right? I think it's the stinkiness. It's how it changes your odor. Yeah,
got it.
Yeah, how it changes the components of how you smell, and that's what they really showed. So, the use of plants and plant compounds can be beneficial for repelling bugs like mosquitoes, but it depends on, which plants you're using and what kind of flower you smell like. Do you smell like a flower or do you smell like a coconut? I guess I want to smell like a coconut. That's, I guess, the thing right now. But I don't know about you, but as we get older, people talk about getting distracted a little bit more often, not being able to focus on stuff. There's research.
I'm sorry, what were you saying?
Yeah, I don't know. I don't know what I was saying. I got distracted by your comment. As the parent of a child who loves to interrupt me while I'm in the middle of making dinner or doing a task of any kind, I lose stream of flow very often. And what was just determined by a study out of the University of California, Riverside in the journal Psychology and Aging, which I will caveat with the fact that they only recruited 19 older adults between 65 and 86. and then 31 younger adults, which probably was easier because they're at UC Riverside, which is a UC research university that has many people in that age group. They looked at the idea that older adults, when they're trying to physically exert themselves, that physical exertion makes them more distractible. And this is, I think, an interesting study. It suggests that, yes, if you're doing something physical, that you're more easily distracted, that your mental effort is focused on the physical effort. In this, though, the way that they did the study, I just kind of wonder, I don't know, well, they had a cognitive task where they had people focus on something and try and do a thing. And then they had them squeeze a squeezy thing. And they were supposed to squeeze up to like from 10% to 30% of their ability to squeeze. So they're supposed just squeezing a ball or squeezing a handlebar and just squeezing it. But then while they're squeezing it, then they added distracting things on the screen while they're trying to be focusing on this other thing that's going on. So it kind of starting to distract, but you're supposed to be squeezing and you're supposed to be focusing. And then there's a distracting thing. And anyway, older adults were more easily distracted when they were exerting more force on the squeezy thing. So what does this mean for aging healthily and reducing distractions? I don't know. It's we might be more prone to distractions. So, hey, young people, if you see somebody who's older doing something like driving, don't try to have a conversation with them while they're driving, because you're going to distract them from the focusing on the driving. I know this is but
if you see an old person on a bicycle, don't go, hey, because that could end badly. Well, I don't know how this gets used. I like that one. Don't distract the driver is always good.
Don't distract the drivers.
I'm curious if this was all on like some sort of touch screen situation also, because
Right.
if you were raised using a screen
Versus not.
you might actually be better at multitasking on a screen because you've been using one your entire life than if you were a person who grew up before these multifaceted screens with multiple windows existed.
Yeah.
So I think that part is tough depending on what the test methods were.
I think also, it becomes the question of how many conversations can you, if you're listening to the radio, can you still have a conversation with somebody and do some other task? Or, what is happening in your life and how much can you focus on? I think it's interesting to correlate this with physical activity. The reason that they're trying to look into this is because people tend to lose muscle mass as they get older. People move less as they get older. And so maybe because of the deterioration of the physical form, that leads to, and this is just me completely speculating, but maybe the deterioration of the physical form leads to more mental effort needing to be spent to maintain it when you need to use it. And so then this feedback effect. But anyway, I don't know, maybe keep up your walking and your weightlifting and you're listening to the radio and talking to people for as long as you can. And young people, don't distract old people. Just be nice.
I also just think multitasking gets harder when you get older, just generally speaking. Across the board.
That may be true. What's also true, though, is that most people who think that they're multitasking aren't. They've done
They're doing everything worse, basically.
No, they
Or
are.
they're doing little bursts of one thing here, one thing here, one thing here, one thing here, and not at the same time.
There was some study done on, good multitaskers were recruited for a study where they were driving and holding a conversation and doing a task. And whatever. And they all thought they did wonderfully. And it turned out their driving was much worse than the undistracted group. Mm-hmm. The reason they didn't notice that their driving was worse than anybody else's while they were multitasking is because they were multitasking. They were distracted. That's the whole, that's what being distracted is not noticing. Like, for instance, I cannot, if I am reading, I cannot hear. I cannot hear the world. Somebody can talk to me. If I'm in that moment, I am reading something, I didn't hear anything that they said. Now, in a room full of people talking, I can kind of track a couple conversations at once. At least I think I can. But in reality, that might be so distracting that I don't even realize that I'm not even catching any of what they're actually saying. People can't tell. That's the whole point of distraction, is that you can't always tell you're being distracted.
Yeah, and some people have a predilection to distraction. So, it's all a thing. Be nice. I don't know what this study actually says about what we can do in the future. Just understand that people get distracted, and that's a real thing. So,
Yeah.
I think we made it to the end of the show.
We did.
Yeah. We did a good job. I would like to say thank you, everyone, for joining us for another show. Glad we could all be here together. Chatters, thank you for being in our chat rooms. Fada, thank you for all your help on show notes and social media. Identity 4, thank you for recording the show. Gord, Arnlor, others who help keep our chat rooms happy, safe places, thank you for being there. And Rachel, thank you so much for editing the show. And as always, I especially want to thank our Patreon sponsors. Thank you to Craig Potts, Mary Gortz, Teresa Smith, Richard Badge, Kent Northcote, Rick Loveman, George Corris, Pierre Velazar, John Ratnaswamy, Carl Kornfeld, Chris Wozniak, Vegard Shefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan, Don Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredos 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, David Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Johnny Gridley, Remy Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Reedy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Adam Mishkan, Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, and Tony Steele.
There's some new names.
There were some new names. I love reading the new names. I hope someone out there wants to be a new name. If you want to be a new name, head over to TWIS.org and click on that Patreon link on next week's show.
We will be back broadcasting Wednesday at 8 p.m. Pacific time. And again, for the second show, 5 a.m. Central European time on Thursday. That's it.
Same time. Same
Different
time.
show.
Same time.
You can find us on the YouTube and the Facebook when we're live. Also, you can go to twis.org/live to catch the live broadcast.
Hey, do you want to listen to us as a podcast? Maybe not while you're driving if it's going to distract you. It's a personal choice. But just search for This Week in Science where podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything heard here today, show notes and links to stories will be available on our website, www.twis.org, and you can sign up for newsletters, maybe even ours.
You can also contact us directly. Email Kiki at Kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twis.org. Just be sure to put TWIS, TWIS in the subject line, or your email will end up stuck to an assassin bug's leg, and they'll run away, and we'll never read it.
Oh yeah, and then but then I guess we're still on the Twitter.
Yeah,
Still there. It feels almost sort of like it's getting to be the end of school, and you're gonna have to say goodbye to all your friends there on the Twitter. But right now, you can still write in our yearbooks at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover or a suggestion for an interview, a haiku that comes through the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views. But I've done the calculations and I've got a plan.
You just might understand that this is the after show. Thanks for joining us, everybody. What a fun time. I'm exhausted. Blair.
Uh-huh.
Oh, you're there. Okay.
I'm
I'm
here.
gonna sneeze.
Did I look frozen?
You did. Are you frozen over there? What's happening?
Do you want to tell me what numbers are in this color test?
No.
You don't?
Ah,
Look at it.
sure.
I can tell the first one.
Oh,
That's the one I can always tell. 12.
I have to put it on a bigger screen so I can actually see. Okay, 12.
This is either a 3 or an 8.
I think it's an 8.
Really?
Yeah,
Oh
it's an
man, I
8.
would have gotten that one wrong.
I was an 8.
10?
29.
No idea. Nothing.
5.
Okay, great. Nothing.
3.
Okay, excellent. 15?
Yes.
Oh, I
That's
got.
so interesting.
I'm 2 for 2. I'm 2 so far. 14?
74.
No, my God. Nothing. There's no number in there.
6.
Okay, great. 65?
45.
Okay. Nothing at all.
5.
Okay. You're lying to me.
No.
Whoa, she's telling you the
Is
truth.
this a 1?
7.
Okay. Nothing.
16.
Okay, great. Nothing.
73.
Okay, excellent. Ooh, squigglies.
Yep.
What number is that?
Is that a number?
It's not? This
No,
one?
that's weird lines all over. I mean, I could probably trace the lines for you, but yeah, not a
Okay.
number. That's not a number.
That's not
There's
a
a
number.
4. There's maybe a 4 in there, but
There's
it's
a
still
4
just
in there?
it is.
Yeah,
weird
there is.
lines.
It's a
Yeah.
This looks like a 6.
You're
think.
right.
Okay, but I don't know what this is.
2. 26.
Okay.
This
12?
is the answer. This is the answer to everything.
42.
Okay. Nothing at all. Squiggles?
Squiggles.
Okay. Nothing.
Squiggles. Yeah.
Okay. Nothing.
Squiggles. Yeah.
Okay. Nothing.
That's a squiggle.
Nothing at all.
Yeah, squiggle.
So
No,
we're
I'm
now
saying I don't even see squiggles in these.
I know. I'm
They'll-
saying there's a
Okay,
squiggle.
okay, got it. This I can definitely see a squiggle in this one.
Okay. That one's an
Ooh.
obvious squiggle.
Okay, great. So I got 2.
Congratulations.
Yeah, so the initial 21 are a form of colorblindness. It gets more specific once you go beyond 21 and you're looking much more directly at the red-green differentiation.
Sure.
Kind of the last portion of the test is sort of different than the So then just being able to recognize that there's something.
So this is the other one. This is the one that truly blew my mind. And we don't have to do the whole
Okay.
thing because it's going to take me a minute, but it's the color arrangement
Onto a big
test.
screen again.
So you're supposed to
match.
so you arrange the colors in order.
Oh. Like along the color
Right.
spectrum.
So like this is impossible for me. This is so hard. it's a no-go is the problem?
I don't know.
Am I doing okay?
You're
I
doing
think so.
fantastic.
Yeah.
This is where it gets hard.
Yeah. I think you're doing great so far.
Well, cool.
Yeah.
Okay. Well, I see now I'm completely lost. These all look the same.
How fascinating. Yeah. I mean, there are a lot of similarities to some of these
Oh, I
colors.
feel
but
like okay, so I feel like this should go here, that should go there.
There you go.
Yeah.
There you go.
And once you had them, you could
I think you
compare
nailed it.
them. I think you got it. Yeah, I'm looking at it, through a screen. We all have to know that, our screens show different colors and there's all sorts of things. But I thought it was good.
Didn't
This
give
is the
you a
art-
result?
This is the art part of how
Wait,
you learned how to
How come
compare
it didn't give you
colors,
the
Blair.
results?
This is the
Didn't tell
result. So it says the thick line describes your order of the test plates. People with normal color order them in a circle, P1 through 15.
Yeah, you were pretty circular there. You did pretty good.
Crossings indicate
good.
forms of color blindness. So the fact I have the protan and the deutan crossing.
Yeah. But that's a minor crossing compared to how it could be. Really.
Mm-hmm.
It could be much
Interesting.
worse. That was actually not bad at all. The one I also found fascinating, used to be at the old Exploratorium. I don't know if it's in the new
Yes,
Exploratorium.
that's where we found out my brother was colorblind.
Oh, really?
At the old Exploratorium, yeah.
Are you talking
Yeah,
about the color blindness room?
No,
No,
Or-
there's this chart.
Yeah, it was a
And
chart.
and so yeah, one person puts there's a color will be in some spot of this. And then you have these two wheels that have the matching color, and so you and your friend can kind of match them up. Now, apparently they discovered color blindness in Kiki's brother there. But what I found was interesting is when you match it up with somebody who isn't colorblind, you can still, because it's a really, like kind of the test you just did, but with maybe 100 iterations of color. Mm-hmm. And so what you find is that I was there with somebody and we were matching up the colors next to each other, but we weren't agreeing on the matching hue to the lit portion of it. And so it that kind of pointed out that we don't all see colors exactly the same.
And
that's definitely don't.
differentiation
Yeah,
is-
that's true as well.
Yeah.
Even
Oh,
in
so,
the color cited, there's a lack of agreement.
Yeah. So Zombie Tom Hanks in the chat room asks what the chance of passing color blindness to children is for women versus men. So I am it's a sex-linked trait. So that's why men are more likely to have it.
They're usually carriers.
On the X chromosome. So basically you only need to get it once for you to be colorblind if you're a man. That's why it's much more likely. To get it as a woman, you have to get both X's with color blindness in it. So it's very, very, very rare. You're more likely to have 11 toes than to be a colorblind woman, which I think is very funny.
That's
Now,
wild.
What's
Okay.
funny is I am currently carrying a boy. He, according to sex-linked trait rules, which genomics are more complicated than that. It's not cut and dried, but according to the sex-linked trait rules, 100% he will be colorblind. Sorry, bud. Yeah, because his X he receives is from me, guaranteed.
Yep.
And both of my X's carry color blindness on them. That's why I am colorblind. Therefore, he has to be colorblind. The question is, because it's actually much more complicated than that, and how colorblind you are is a whole complicated thing. It has to do with the chromosomes and like exact mixing and all these things. So anyway, how colorblind he is, it will be a question later on. But he will be, at least technically, somewhat colorblind.
I have a question.
Sorry, bud.
I have a question for you that I totally forgot
Yeah.
about. In relation to the case study. Now, we know that it takes a while. Like I think the first time you used the Enchromo glasses, we were 45 minutes in or something like that. It was a while before you started to see the effect.
Yeah.
And then you now tell me it takes a week for it. You have to use it for
full,
a week before
for full potency.
For full
Yeah.
potency. Question.
Yeah.
When you take them off,
Yeah.
does it immediately go away?
That's a great question. Really difficult to say, partially because the sunglasses work way better than the inside glasses. And so you are dealing with a brightness issue when you take your sunglasses off. So it's actually pretty hard to tell. I would say if I had to make kind of like a gut response to your question, it sticks around a little. It's not the same, but it definitely, like, you continue to notice things a little bit, slightly.
Right. So the brain
I haven't
is
worn them in a
still-
long time because I can't be trusted with sunglasses. I destroy them, and so
just taking care of them.
yeah. So
same.
they're getting a break right now, and I take them out for special occasions or like hikes or stuff like that. But I have my $15 Target sunglasses is what I usually rock these days because that's what I can be trusted
Go
with.
through them, and it doesn't matter if they break.
Fall off my head. It's a whole thing, but
Yeah. But if you've ever worn like
Yeah.
any tinted glasses, you know, there is a certain amount of adjustment that our eyes make. So if it, you know, the light changes the amount of blue or the amount of green that's getting into your vision, then that opposite part of the spectrum is heightened. And so when you take them off for just a little bit, everything looks, you know, a little bit more whatever that color was.
Right. So when
And
we
so-
talk about it a little bit.
But that happens a little bit. But if you wear it for like a week, like Blair was saying, what's happening is if your brain is actually shifting, then that is going to have more of a long-term residual, right?
Yeah,
because your brain
because-
has to switch back.
Because the interesting part to me here is, and I'm very curious. Next time you're using them for a little while, take notes when you take them off.
Yeah.
I think I remember you reporting that it kind of went away, but orange still was loud or something like this, right? Because the way that the Enchroma glasses work is, you know, I talked about the overlap of the red-green spectrum being confused, and so the brain just identifying it as one thing.
yeah.
The Enchroma are largely designed to block that middle portion of the overlap, so that they can be
Separate.
perceived.
They like force them to hypercolor, basically.
Yep.
Yeah, they perceive they force them to be separate wavelengths, and so then the brain begins to do the processing.
That's definitely
When
how
I'm-
it kind of felt to me, was like looking at a blacklight poster almost. That's how it felt when I was wearing them.
Okay, and so, but then what I'm really curious about is that plasticity in the brain that has now been identifying these separate things, to see how well it can maintain it after the glasses are removed.
Right.
For how long? Is it 15 minutes? Is it a day? Is it that following week?
Is it this one guy?
How
But
long
again,
would it
I also-
be? Yeah.
Also, in looking
But
at this, I found all of this hate commentary for Enchroma, and all these other different techniques, and they all came from folks who have like the worst kind of
The color
of
blindness because
blindness.
it's not helping what they're
because they're not in that in between range. They're not having
Right.
an overlap problem. They're having
Well,
a
and if
tone
you
missing
take
problem.
the test on their website, they'll tell you these glasses aren't
They
for
will
you.
tell you, and they
But I
have a
think-
money back guarantee on top of it, just because
Yeah.
we're not even. It
didn't
sounds
work.
like we're doing
Here,
a promo.
Send it back.
And I also think that a lot of times people buy them for people as gifts, and they don't know what kind of color blindness their
That's right.
significant
Probably,
other or
yeah.
friend has. So they don't
Yeah.
know any better.
Yeah, but just for those people, the magic mushrooms might not work for you. The Enchroma glasses might not work for you. Nothing will help you. But that doesn't mean that there aren't people who can.
But we
still-
don't know that. The mushrooms might work. We have a sample size of 1 so far.
Sample size
Yeah,
of one.
and it's not even. It's not even. Well, technically, it's a somewhat controlled study. One of the funny things that was in there too was that the. I can't remember what the time frame was. It was like, you know.
One
2
time
weeks or
in
3
college.
weeks. 4 weeks later or something like this, they weren't at the peak on that test that they reached in the 24-hour after period.
This
but
is
they
for
were
sure
still-
a bunch of researchers did shrooms.
Yeah.
And one of them was
see
like, I can see colors, guys.
Yeah.
They were like, we
They
should
should
write
have
a
just
paper.
called us and said we're going to share this on the show. But but they reported also actually that the effects in the case report they kind of focus on, you know, like 4 weeks later, you know, they're 4 months later, whatever it was. I can't remember.
40 weeks.
They still had the effects. 40 weeks was the
Stem
mouse.
cells.
That was the stem cells, yeah.
But they actually
cells,
reported that they still
mushrooms.
had improved
Potato,
color
potato.
vision.
Rub a mushroom on it. It'll be fine.
They stated that the individual did report still having improved color vision a year later. Not at the height, not near the peak, but still better than it. But hang on, asterisk. But they had used multiple psychedelics in that period. Therefore, this is ruining
This is what I'm
the study portion.
This is a bunch of
the-
researchers who
Quote
like to do like LSD and shrooms. That's
Quote
what this is.
unquote study.
Study,
This isn't
yeah.
a study. Yeah,
No,
this
it's a
is-
case report of somebody who did a self-study. I would still call it a study, but because they used colleague, that's the only thing I'm grabbing onto is they didn't just say, hey, this guy walked up and told us this story, so we did a case report on it. They had a
this is bunch of
colleague who they
scientists who like to
trusted
do
enough
psychedelics.
to
Good on 'em.
do a self-experiment that they would report on it.
That's
I
okay.
still have
All right.
questions. We
No,
need more research.
Also though, here we have here's a perfect example of an illegal substance that doesn't have all of the negative effects of these drugs that doctors are prescribing multiples of without a study behind. So I'm just saying.
Well,
in the
Justin,
fairness
you
of
don't know
pharma-
You
that
don't know,
because
because
there
there's no
aren't
studies.
studies of mushrooms versus XYZ. So
Or
you
with
actually don't know
XYZ.
that. Yeah. According to your rule,
Technically,
that
you're
shouldn't be
correct.
allowed.
Technically, you're correct. However,
And technically, I don't know.
However-
I've ever heard that psilocybin is addictive.
oh,
But
I gotta go to
however-
psilocybin rehab.
Yeah.
Oh yeah. Oh, I gotta get the psilocybin toxins out of my body. No, there's no such. There's no
No.
such.
I mean, maybe for this guy who suddenly can see colors.
Maybe he
There's a-
should. Yeah, maybe there's something there. I need to see dreams.
Yeah, let's go.
Oh, okay.
bed.
In that case, say goodnight, Blair.
Good night, Blair. Say good morning, Justin.
Good morning, Justin. Good night,
Night,
Kiki.
Kiki.
Good night, everyone. Have a wonderful week. We'll be back again next week. Looking forward to all the science we get to share. Stay safe, stay happy, stay wherever. Stay curious.
Stay psychedelic.
Stay. Indeed. Whoa, the colors, man.